We're about to start, so please put on your seat belts because we're going to accelerate now. So first of all, just want to thank the audience. The questions have been terrific. Keep them coming because that's why we're here. This is not a traditional, one talk after the next. We're really expecting a lot of discussion, audience interactions with the speakers, but also with themselves. This is really the spirit that we have here. Okay, so the next speaker is Igor Timofeev. So I have a personal story to tell, which goes way back to a talk I gave at NYU. And it was a time when gamma was really hot because of Wolf Singer, who had measured gamma in primary visual cortex and thought it had something to do with consciousness. The trouble with that experiment was that it was done in an anesthetized cat. But it was nonetheless fascinating and interesting from a mechanistic perspective. And I at the time had a postdoc who developed very detailed models. This is Bill Litton, of inhibitory cells, the basket cells, that could create very nice gamma because they had access to the soma. They're primarily the synapses are on the soma, and you have a very fast reaction, very fast feedback, negative feedback, to give you a nice burst of gamma. And you don't get that if the inhibition is on the apical dendrite. So, it gave us a hint. And in any case, I get a call from Mircea Steriad. Who's he? Well, it turns out he's at Laval, and he's one of the masters of recording intracellularly from cortical cells during sleep. I think the record is like 4 intracellular recordings during sleep. And he said that he was interested in my talk because there's inhibition in the thalamus that produces sleep spindles. And was I interested in collaborating? I said, sure. And so that started a collaboration that lasted 15 years and produced 30 papers. And then a book eventually that came out with Cambridge University Press with the Landis Decks. But in any case, we continued this. During that period, Maxim Bazhanov was a postdoc in my lab, and Igor was a postdoc with Mircea. And what really fueled this collaboration was the two of them getting along because they both spoke Russian. They formed a close bond, and that's the way science works. It's a social enterprise, and it really depends on people collaborating who really enjoy working with each other, and it's amazing how that impact that could have. Okay, so who is Igor? So Igor has really carried on this tradition. He's carried on the tradition. By the way, we were working on sleep spindles back then, but he's continued that tradition of what's going on in the cortex during sleep memory consolidation, and so forth. He's also, as you're going to be hearing, I think, seizures, has gotten interested in the mechanisms underlying epilepsy, which is a very complex disease. It's very heterogeneous. We've collaborated, my lab and Maxim and Igor have collaborated over the last at least 10 years. Maybe it's longer, 15 years? Oh, okay. Yeah, well, that goes, okay, so that we're talking about like 25 years. Okay, so this is an ongoing collaboration between our three groups, and this is knowing the neural mechanisms underlying these different brain states and abnormal brain states really has propelled, I think, our understanding of what is driving things like memory and how does epilepsy, epileptogenesis in particular, how does it develop? It's not overnight. It takes many weeks. And so this is something that Igor might be telling us about.
Thank you very much for the introduction. I just want to make one comment related to Yuri's talk. I collaborate quite intensively with two computational neuroscientists, Terry Sinovsky and Masim Bajenov. And every time I come, if there is a barbecue, it's a very decent barbecue. It's not, yeah, so. Yeah, so here are my affiliations. Once I brought my daughter to my talk, and she's an artist, and this is what she did. Yes, so and I work in Quebec City, which is here, and this is, yeah, I believe that your current government will offer us custom free travel, right? This is what it is going to be. Yes, so, and I'll start from brain states and where is my, yes. The first thing I'll show you what cells are doing, I'll stop, during different brain states. So here is the EEG, it's not classical EEG, it's LFP, but we did it for YouTube, so it says EEG. Intracellular activity. This is electrocoologram. If you see anything now, this is artifact because these electrodes are located close to the brain, and so it picks up some brain signal, but you will see when eyes are moving, it will be different signal. This electromyogram, and you see these little spikes, and in cat, during slow wave, yeah, this cat, yes. During slow wave sleep, these spikes are continuously present. It's not the case for the rodents because very often the muscle tone is very, very low during slow wave sleep. So the cat sleeps, and then it is going to REM sleep. Here is transition. So first thing you see, those little spikes disappear. Then this is the last hyperpolarizing event that occurs during slow wave, and it's a stable member potential and no slow waves, obviously, in the EEG. Well, I don't know, I'll click here, yeah. So these are rapid eye movements, so it moves. And then you'll tell me when the cat will wake up, because now it has dreams, a tasty mice, whatever. Now, yes, now. So why? The most obvious difference are the muscles. It's not much difference in the membrane potential of cortical cells, not much difference. There are some subtle differences at the level of the EEG. Yes, so to repeat, slow wave sleep, we have these hyperpolarizing potentials, which are called down states and up states. They generate slow waves in the EEG, or LFP, whatever. They're absent during REM sleep, they're absent during wake, and during slow wave sleep, the muscle tone is stable. During REM sleep, it is absent except some twitches occasionally, and it's obviously variable during wake.
So Igor, it's worth mentioning that the up and down states were discovered by Mircea.
Well, first of all, with all respect to him, I would say he never called them up and down states. He calls them depolarizing states and hyperpolarizing states. And the main reason when new terminology up-down appeared when cortical cells are going to the up state, in primary thalamus, there are typically spindles, which are hyperpolarizing events, and you can call them active states because they're active, but they're not up states.
Was Johnny Rizzo?
Yeah. Who called first? Yeah, I think 96, right? It's the striatum, yeah. Yeah, but Mircea, I mean, he never, if you read anything, what he wrote, he never used up and down state.
We can forgive him for not knowing that. He had the data. He had the electrode recordings, and he correctly recognized they were hyperpolarizing, depolarizing states that were somehow global.
Yeah, yeah. Yeah. Yes, so those are three basic states. Unfortunately, the life is more complicated than that. So here we're looking on the marmosets. We use these neurologgers. It's a small thing, small battery, and memory card you can implant in the head and have 24-hour recording or nearly 24-hour recording depending on your battery. And so this is what we see. The first slide I show you, this is wake. The whole thing is wake. We have several electrodes in the different cortical areas, motor, frontal, whatever. And these few electrodes, we have this spectrogram. What is absolutely remarkable, well, first of all, we do have some events which correspond to classical description, some gamma bursts, some beta bursts, but here we have enormous mu rhythm. It's just something remarkable. You can find it here. The mu rhythm classical description is when the animal does not move, does not see movement, does not think about movement. And it is recorded in somatosensory and motor cortex. So this is exactly what we have, this motor somatosensory cortex, this enormous mu rhythm. We have all kind of other things like in hippocampus, we have sharp wave ripples, so everything else is another feature of this mu rhythm, that it goes kind of in alternation with alpha rhythm. Alpha rhythm is more occipital. You can find some alpha frequency everywhere, but it's more occipital, and when alpha rhythm is strong, mu rhythm is low. When mu rhythm is strong, alpha rhythm is of low power. And I will show you, yes, there are beautiful ripples, you can see them without filtering, yes. Within mu rhythm, this is mu rhythm episode, and they are just, you don't need microscope to see them. They happen there. And I'll show you what the animal is doing during this mu rhythm. You see, it does nothing. It just, there's no movement, it doesn't move, and it's like that. So we have obviously different brain states because the activity, the LFP activity is different, but it's all wake, right? It's all wake. So it's much more subtle than being awake or being asleep. Now, sleep also will have the same kind of electrodes, and obviously have slow waves, we have spindles. During REM sleep, there's a lot of beta activity, very strong in the hippocampus. We, as Yuri mentioned, in primates, we do not have theta rhythm, but there are some theta segments. There are 3-5 theta waves that happen, but it's nothing like stable theta rhythm. There are obviously sharp wave ripples, all that. So the key elements are slow waves, spindles, but everything else is also present during this slow wave sleep. I believe the next, yes. I will show you how brain activity is different. So we're looking on the animal with this blue head, blue protector. So it's obviously sleeping. Now we have these slow waves and spindles. It's sleeping. Wakes up, checks around, goes through mu rhythm. So here is a line. And go into slow waves. Spindles, slow waves. Yeah. So this is, again, within very short segment of time, we saw two big rhythms, slow wave sleep, wake, slow wave sleep, but we saw how variable these states are, right? It's not a homogeneous wake, not homogeneous sleep. Now, we try to quantify a bit these sleep states in marmosets. Here is one sleep cycle. Well, it's a bit more. Marmosets do have sleep cycles, about 12 to 15 sleep cycles per night. Let's say starting from REM, could have some brief wake, drowsiness, light non-REM sleep, deep non-REM sleep. So here, the way we quantify, first we plot delta power, so we segmented, I believe it was 5 seconds segment, and plotted these 5 seconds of delta power versus muscle power. And then we shift by 1 second, so 24. So this is 24 hours, each dot is 5 seconds, and this overlap with neighboring dots. Yes, and then it's delta power and sigma power. Sigma power is where this, mu rhythm is present, or basically sleep spindles are also forming there. And then, based on that, we use self-organizing maps. It's an artificial intelligence kind of approach. Someone told me that only lazy doesn't do it, so even if I'm lazy in that, we did it. And so we clearly see separation of states in this self-organizing map. So there are different states. What is very nice, in these different states, this is 24-hour. Of course, battery does not always last for the whole period. But we had 3 animals in the same cage. Rat is awake, and so you can see that most of the time during light period, marmosets are awake. They have some naps, typically around 9, 11. Yes, in the group of 3, they had a community. That one was guarding. So when this animal had nap during the day, that one basically did not have a nap. But it was more tired, and at the end of the day, it started to sleep more, it started to sleep before the other animals were sleeping. So there's also this interaction in the group that should be considered when we look on these events. Yes, so mice have very fragmented states. This is what this muscle power, this is delta power, yes. And during light, we can see that mice are continuously either awake or sleep. No, this is delta. So this awake, this sleep, this is wake, because this is muscle. Muscle high, and we can also detect REM sleep. And when we look on all this together, so here in this study there was comparison of old and young animals, so forget about that, just look at that. So during light period, this is one animal night, well, days, 24 hours. These are animals, each line is average for one animal for over months about, etc. And this is total average. So we can see that about 30% of time, 40% of time during light period, mice are awake, and about 50-60% of time they have slower sleep, and then they have REM sleep. And then at the beginning of night, so they become more awake, and they sleep less. So this common confusion, well, if you do it, you are not confused, but this common confusion that mice are awake during night and they sleep during day. No, they are awake more, they sleep more. But it's not black and white, because again, I come back, it's very, there's nothing stable. It's very fragmented. Then there is this. So I told you already about slow waves and other kind of activities. Now, these are 3 traces. The green is that one, the red is that one, the blue is that one, and that one below is a muscle tone. You see these little spikes, it's heartbeat artifacts. So the neck muscle electrode pick up heartbeat artifacts. So what is the state? You see, those slow waves, and basically no muscle tone. Beauty.
We have something like this with electrodes that are 500 microns away. You look at the unit activity, you can say there is an up and down
Yeah, yeah.
Brain is not globally synchronized.
Yeah, but overall, would you say it's slow wave sleep, more or less?
It's certainly the animal is not running, but
Cool, agree, yes. That, again, no muscle activity, beautiful theta rhythm.
that's running.
No muscle activity at all.
Then REM.
REM sleep, perfect. So this is the same what I showed you before.
It's the intermediate.
You have this area where you have huge slow wave activity. You have areas where you have beautiful REM sleep, formally defined, and you have areas where it's kind of a mixture. Well, I'm not sure about intermediate because, so by putting arbitrary thresholds, we identify all this epoch as a REM sleep, and that segment that I showed you just before, it's right in the middle. So there's a minute or so, well, this is 25 seconds, yeah. 100 seconds of REM sleep, and right in the middle is that. So we do not sleep or awake at the same way in different brain areas at the same time. Well, I'm not sure about us, but about mice is like that. Yeah. Yes. So I call it Christmas tree. So blue is slow sleep, red is wake. And so we identify kind of state per electrode. So we do not look on all of them. It's slow sleep, and then this, and then. Because Christmas tree, it has red and blue and green, and here it's two. And then, now it goes, that was muscle jerk, because everything was away. And now it goes to everything you want, right? Okay, yeah, so this is the way mice sleep. So for this part, conclusions, there are three main states of vigilance, the way you know them. It's wake, non-REM, and REM, but there's more. Each of these states can be subdivided on as much as you want. You can say wake is active wake when we're performing, doing whatever. Typically, it's gamma beta activity. Relaxed wake, it's alpha rhythm. Relaxed wake and drowsiness, it's mu rhythm. It's all wake. We're not sleeping. Non-REM sleep, well, in human, it's more or less well done. Now there are three states, stage 1, stage 2, and slow wave sleep. Before it was 4 stages. REM sleep, there's active REM, where you move eyes, and there's passive wake, when there's REM. When there's REM, but you do not move eyes. It's still REM sleep, but you do not continuously move eyes during REM sleep. Yes, and then several brain states can coexist. Yeah, so for sure, in mice, brain states are fragmented. And in marmosets, brain states, and in human, brain states are consolidated, meaning the marmosets sleep the whole night. Like us, they can have a little wake up, but they typically sleep. And the day they're typically awake. Yes. Neuronal excitability, because I had 3 points. Actually, I added point number 4. Maxim, could you tell me 10 minutes before, please? Yes. Yes, so excitability. So these are 2 slow waves. It's done under anesthesia. It doesn't matter. It's just to ask question. What could it do in terms of neuronal plasticity? What we know that if you stimulate with 1 Hertz frequency, you induce long-term depression. And the sleep source oscillation is about 1 Hertz. Now, within this sleep source oscillation, you have cells that fire, and they fire with quite high frequency, right? So, and if you stimulate structure with high frequency, you induce long-term potentiation. So, what you do, because there are 2 things are possible: slow waves, but within each slow wave there is activity. I'll skip this. Yes. What is also important, I believe this, actually I didn't, I'm sorry, I didn't put, it's Hestin, Galaretta Hestin 98 or 99, Nature Neuroscience. They look on, I'm very sorry, so it's done in slice. No? Isn't it slice? Yes, so they recorded connected cells, and first they stimulated one pulse per whatever, 10 seconds, and they have some responses. And then they went with wake kind of stimulation, 20 hertz, presynaptic, and they observed steady state depression, because the synapses cannot maintain for long this kind of stimulation, 20 hertz. Then when they stop this rebound, and again, single individual stimulus produces that kind of response. So what it tells that when your network is active, it is depressed. And this is true for excitatory, for inhibitory, for everything. Yes. So on basis of that, we try to look how brain response is affected when there's an activity, stable activity, which is wake, more or less stable activity, versus sleep, where there's this up and down state and all that. So we took a cat, and so first I wanted to see if you stimulate cat fur, how would it respond in different states. Impossible to sleep, because every time you touch cat, it wakes up. So it's just impossible. Then, so this is somatosensory system, very simplified. Peripheral receptors, dorsal root ganglia, dorsal column nuclei, and this is VPL of thalamus, motor thalamus, somatosensory thalamus that goes to the cortex. So we put stimulating electrode pre-thalamus, before thalamus. So we skipped one synapse in the major pathway, but still cat does not wake up in that. And so all this is to show you that we formally detected states of vigilance correctly. We have here wake, slow sleep, REM, wake. So this phenomenon that I'm going to explain you, I saw it, I just was looking that response was increased after sleep. And I didn't understand, because there was dominant idea of Tononi that it goes the other way, that sleep down regulates everything. They have plenty of beautiful indirect data, and there's nothing direct that support all these claims. So in order to be sure that we will see this phenomenon clearer, I came to the lab, I played for 2 hours with the cat to be sure that it does not sleep. Then we fix it to recording system, and we measure evoked potential. We look on the initial response, the first response, the synaptic response that is given. This is that. Then cat went to sleep. Well, normally we do not stimulate somatosensory system during sleep, so we stop stimulation. And then this initial response was increased in the amplitude, you see. And it stayed quite increased. Wow. Then there was next sleep episode. We just decided to keep stimulation just to see what happens. As expected, depending when you stimulate up-down state, the response was variable in amplitude. But then again, the last wake, it was increased. And here, these are all animals that we made. So if it's 1, then there was no change between wake 1 and wake 2. In this, so wake 1 and wake 2, I mean that there should be at least 15 hours before experiments in the same animal. But then the response to the second stimulus was continuously increased. And this is for everything we're doing. Cool. Then sleep increases, responsiveness is increased after sleep, synaptic response. Well, this is not yet synaptic, it's a work potential, could be yes, so that was done with Sylvain, Sarah helped, and then this is what they did, yes. Okay, I have 10 minutes, cool. Now, to confirm that these are synaptic responses, we obtain intercellular recordings. Again, the idea is to get a cell to be in a wake state, to stimulate, to estimate, these are responses, then to go to slow wave sleep, and then to record another wake, and those are the responses. Of course, you can see that they are different in amplitude. So after period of sleep, the synaptic responsiveness is increased, and we got 6 cells. It's very difficult to do technically, and in 5 out of 6, there was this increase in the response amplitude. Good. So how does it happen? Why it happened? So we went to look on that in slices. So again, we start from natural conditions. We took some recording of local field potential in the wake cat, detected spikes, then patch cell in a slice, and stimulated with a pattern that was recorded during wake, right? So this is shown here. This is normalized because many cells. So initially we go with 1 pulse per 10 seconds, like typical classical in vitro. Then here, 10 minutes of real wake kind of stimulation, I mean real spikes that we recorded. And then after, there's not much change, right? Then we thought, okay, during wake, there are plenty of neuromodulators. We would add a carbachol to activate cholinergic receptors. We did, as expected, the amplitude of synaptic responses was decreased. So here is before carbachol, it's 1 per 10 seconds. After carbachol, it decreased. Then 10 minutes of wake stimulation, and then boom, some very short-term facilitation, and then it goes down. Not good. Then we went to sleep. We recorded sleep spikes. We also recorded slow waves. And in vitro, in slice, so we stimulated spikes like during slow wave sleep, and we hyperpolarized cells just to replicate slow wave pattern. And so what we observe, if we're doing just synaptic stimulation, no hyperpolarization, we obtain very short-term facilitation. But if we combine synaptic trains, wake synaptic trains, with hyperpolarization, we obtain long-term facilitation of that response. So slow wave sleep increases excitability, synaptic excitability over very long period of time. Could we increase synaptic excitability and increase memory in mice? So what we did, we have recordings of slow wave sleep and wake. Again, many electrodes in different areas. Wake, delta power is low, slow wave sleep, delta power is high, so this is clear. And then optogenetically, we stimulated frontal cortex, which receives inputs from ventral hippocampus. And so each stimulus triggers slow wave during slow wave sleep, but many trigger during wake. So if there is a wake, the only time when they were unable to trigger slow wave during wake was when there was muscle tone increased. Muscle tone increased, you have some short response, but not a slow wave. And as you can see, so we'll start. So we stimulate frontal cortex. This is backward visual. Slow wave sleep, wake, slow wave sleep here. And more you go to frontal cortex, here it's the delta power is the same. So the structure that receives ventral hippocampal input is in continuous kind of sleep, right? So we just improve it. So what they did there, we took mice. This is a novel object recognition test. It's not novel object location test, it's a recognition test. So we have two objects, and then we change one. We train the animal to learn these two objects, and we have groups of animals, control animals, and the ones that we stimulate with optogenetic. So both animals spend more time with the new object. So you replace object. Yes. So what is stimulation? Stimulation is this optogenetic stimulation in the first 4 hours of dark cycle. So meaning when they are mainly awake, we just stimulate them more. And the experiment to measure behavior is done within an hour before the onset of light cycle. So when they normally have to go to sleep, we just test before. So both stimulated and non-stimulated had similar response, similar interest in new object, but stimulated lost interest to the old object because they learn that that thing is something boring, I know it already, and they didn't go to see that object. Other parameters that we measure they were mobile, they were travel the same distance, so we did not affect their behavior, we just affect their learning ability. 5 minutes, Maxim, yes, no? I don't know, yeah. So, what did I tell you? That neuronal excitability after sleep is increased, as kept intrinsic excitability is long, and increase in excitability after sleep is likely a factor that contributes to the memory consolidation. I'm not saying this is the only factor, but it just that helps. Now, transition to seizure, because again, so sleep increases excitability, and seizures are increased excitability. It's the second side of the medal. So we published that long time ago, and that is in cited, and I can tell you it's all wrong. So these are slow waves, and we see intracellular recording that corresponds to slow waves. This is a seizure, and again, intracellular recording clearly corresponds to the seizure. And we see this involvement from slow wave activity into the seizure, right? And the conclusion was that sleep progressively is transformed into a seizure. So, well, electrophysiology does not lie, so it is not wrong, it is like that. But the thing is that that is done under anesthesia. So when under anesthesia, we see it very commonly. Ketamine cell is in anesthesia. If you keep it for a few hours, animals start to do seizures. Now, we have done multiple species, cats, mice, marmosets, with undercut, cortical undercut. You different part of the cortex, here is a knife, and then animals become epileptic. So what we see most commonly, if we do that, is that the differentiated hemisphere displays more of sleep, while control hemisphere, not affected, is more awake. It's kind of, this is also known for human. Of course, nobody is doing undercuts in human, but brain lesions or all that could lead to very similar phenomenon. Now, what is important? This is a marmoset. With sleep, we see slow waves, spindles or sleep. We never saw any, those are slow waves, but they're very much before the actual onset of seizure. So there are no slow waves at the onset of seizure. The animal is asleep, the spindles might be a bit, and then the onset of seizure start to cease. I'll show you. Of course, that was night, and they slept in the cage, and for the intimate behavior, we did not put camera in the cage where they slept. But next day, this is the same animal. You'll see how the seizure looks. Of course, here there was no battery, so we could not record electrophysiology. But it's the same animal, and this is kind of seizure that is produced by the undercut. You believe me, right? Yeah? Even if you're a computational neuroscientist, this is how it looked like. Well, I believe it's something behavior adaptation because they live in trees, right? And if anything happened, they would fall, they would be killed. So it's possibly something like that. Of course, our marmosets do not live on trees, they live in a cage, yeah. So again, it just to show you that in cat you can have this typically starting seizure activity, paroxysmal activity during slow wave sleep, and it can even persist during REM sleep, which is not very typical in human. Yes, so mice. It's one of the last. So slow wave sleep, right? Slow wave sleep. It's the same mice with undercut. Some jerks, yes, slow wave sleep. So you see it's clear slow wave sleep. Okay, it's just long that you see that it has slow. And then slow waves disappear practically. It's still sleeping, but no slow waves. And the seizure starts. And this is seizure number 1, and there will be 2, and then there will be grooming, and there will be. So again, in mice, like in marmosets, like I saw then in cats, seizures do not involve from slow wave activity. Involvement from slow wave activity, it happens when there is anesthesia. No anesthesia, not such a thing. Yes, here we play with neuronal excitability. So it's undercut. When we do undercut, tissue is differentiated, excitability goes down, and then it progressively increases, and the animal becomes epileptic. So we injected dreads, hyperpolarizing dreads. Around the area, they reproduce in cells. Then 2 weeks after, we did undercut, implanted all electrodes. And if we inhibit this area around undercut, it boosts seizures, animal become epileptic, and this is 30 hours after undercut. That particular animal died here. This is the last kind of segment of recording. Now, in order to compensate for this hyperexcitability, we injected here excitatory dreads, the depolarized cells. So in order to kill this, so this way, into the slow wave sleep. So where we inject, there are slow waves in frontal cortex, in somatosensory mode, but where we do undercut, the slow wave amplitude is very, very, very low. So we essentially prevent slow waves to occur. And these animals did not develop epilepsy, did not develop seizures. This is sum of all experiments. It's control. Undercut, how many seizures happen per day. Undercut with inhibitory drugs. It's not all animals that die on 30 hours, but some of them did. And when we excite this area, this traumatized cortex, essentially we kill epileptogenesis. So I strongly believe that neocortical seizures are nocturnal. Nocturnal related to sleep. Nocturnal does not mean evolving from slow wave sleep activity. Not all seizures are it's exactly the same animal that you, no, it's a different animal, but the same procedure that was done, the cortical undercut, and here is that, yes. So what did you see? It's the last slide. Yeah, I know, just, yeah, I play it again, yeah? What did you see? Cool, yes. How do you know it? Because it could be just scratching artifact, right? Ah, yes, yeah. So exactly, Yuri is absolutely right. Look at this. First, you see it start to scratch, and you see EMG activity, but no seizure yet, right? And then this seizure activity in occipital cortex, which has nothing to do with scratching. Yes, so neocortical epilepsy is essentially nocturnal. It's not necessarily driven by slow wave activity, although on a large scale, slow wave activity modulates excitability. But it's not that slow waves would drive the seizure. Yes, and if we play with neuronal excitability over a long period of time with dreads, we can control development of this epilepsy. Yes. Yeah, I wanted to show you how brain stimulation can help, because, of course, in real life, we cannot take a soldier to say you will get a bullet in the left hemisphere, let's inject dreads, and this cannot be done. But with brain stimulation, we can play. Yeah, please, comments.
Okay, thank you. So, there are humans with epilepsy where they implant electrodes and stimulate them, which is a way to alleviate. You have to predict it's coming, and then you stimulate the cortex, and it does alleviate, but it looks like you have more evidence that just depolarizing.
Well, yes, so this is absolutely
If you have a question,
different time scale. This is absolutely different time scale.
Oh, I see. Okay.
Because what you're talking, stimulation in human, this is something that within seconds, a minute, something of that kind. Here we're talking about stimulation in order to never to see what will happen after. So you stimulate and there will be no epilepsy. We do have, so with transcranial direct current stimulation applied during slow wave sleep, we can modify slow wave activity over, well, we did it 4 times, let's say, over 90 up to 100 days. So it's really very robust. We can increase, we can decrease. Unfortunately, we do not know yet parameters whether this would work for everyone, because you take one animal, you do that, you have that result, long, and you take another animal, do exactly the same, you have a different result, and again, long. So this we're trying to figure out what is there.
Okay, yes. So next question.
How do you give these marmosets epilepsy? Did you buy them that way, or what type of epilepsy do these marmosets have?
Well, the one that I show you, it's a typical tonic-clonic. So the first you saw it was starting with 8 hertz, and there was muscle tone, so which is tonic epilepsy, because when the tonic-clonic, it's a behavioral thing, could be measured by muscles. And then there was clonic because there was muscle jerks with 2 hertz frequency, so it's that kind of epilepsy. Well, we do have some examples. It's all developed in time, right? After undercut, you may have acute seizures or not. This is not epilepsy, just response of brain on trauma. Then for weeks, there is nothing. And then you start to have a subtle kind of absence. Sometimes, you have 3 hertz, and it does nothing, and then it start to go. That was one of the, well, actually I don't know about you, our animal protection, when they see epilepsy in marmosets, they want to kill marmosets. And when I presented to neurologists, they say, what do you do? I say, I kill them. Why? Because this is the way we protect animals, that they would not suffer. And they said, look, our patients would be very surprised if they would come, with epilepsy, and you just kill them. Yeah.
But are you buying epileptic marmosets, or do you?
Oh, no.
Buying normal
It's just undercut. And I show it's not only marmosets, it's the same cat, the same thing in mice. The only difference, critical difference between mice and other, so in cats and marmosets, you do undercut. There are acute seizures, disappear at 12 hours, roughly. Not necessarily present, and then there is a month or so silent period, no epilepsy. And then it starts to be, and then, so what we want to do is to control that silent period, not to get there. In mice, there is no silent period. You have acute seizures, goes down, but the next day you start to have continue. So there's no silent period that we can do something to control in order to translate this then to patients, right?
Yep. Are these are wild type monkeys and marmosets?
Well, it's a wild type. So they are not taken from the nature, they are taken, they reproduce, but this is not genetic model, let's say. Because I don't know for the reason, but what they call zero gene, I mean, the one that you pick up in the forest, they impossible to work. They're impossible to work. I never did it, so I cannot tell you more. And starting from the first generation in animal facility, they become workable. This very difficult because they knew my postdoc and a technician. And cage is empty. There are 3 animals there. When I enter, I never saw them. There's nothing to hide, but they hide. Right? So it takes months that they adapt to be with those 2 people. Yes, it's not easy to work, but results I like.
Okay, next question.
Yeah, really interesting talk. I had a couple quick questions. The first one was these mu rhythms you see, this kind of drowsiness state that seems to precede the slow wave sleep. Do you see it before every slow wave sleep bout? Is
Yeah.
it like a consistent
Well, I wouldn't say 100%, but I would say 90% to my eye. Well, I just don't remember because we even quantified this in the paper.
Yeah. Okay. Got it.
Yeah. Yeah.
And then the second one was so it was interesting seeing these like whole cortex recordings where you saw that the different brain regions were in different kind of sleep state or sleep wake states. Do you see any kind of, when you look at those, let's say a REM state, a REM-like state within one small brain region when the rest of the brain is in the kind of like a awake state, do you see differences between that and the kind of highly synchronized states, or are they almost like visually identical?
What was striking, the very lateral part of somatosensory cortex, there was almost never REM sleep that we could formally identify there. It's not 100%, but it was. And then one of my previous postdoc, he published paper on sleep and different kind of sleep in mice. And I asked Sylvain, so why didn't you tell anything about REM sleep? I mean, slow sleep, coherence, whatever they measure. He said, so before your paper, we were not sure because we had most interest in somatosensory cortex. As we did not see REM sleep, we did not understand why. And then there was a recent paper, it's, I believe they measure calcium or something, and acetylcholine. And apparently that part of the cortex does not receive much of cholinergic inputs.
Okay.
So there's a morphological basis for that, yeah. Again, I cannot tell it for every kind of animal because we're all different, right?
Yeah. Thank you.
Thank you for a great talk. Quick question about, so when you have a continuous epileptic episode, how do you determine epilepsy 1 versus 2, event 1 from event 2? Is it there a change of state in the sleep cycle? For example, they go back to, versus like it's emanating from a different region of the brain?
So the really bad part of my talk that I did not give you much quantification of seizures because they are very variable. They occur rarely. Let's say in marmosets you have 1 or 2 per day, sometimes 1 every 5 days. So these are epileptic animals. Yeah. But so it's very difficult to develop a tool that would pick up, because and every time when seizure happened, it's a different kind of seizure. It's a different, So for mice, we were okay. I mean, what I show you, quantification, but that is a real problem. And when it goes from slow oscillation to epilepsy, there, although I did it a lot under anesthesia, I cannot put my finger and to say this is already seizure. I mean, there's a few seconds where if you convince me this is slow oscillation, yes, you say it's a seizure, yes. Definitely there, it's not seizure, there it's seizure, but transition is very difficult to define. Yeah.
Okay.
Okay, thank you. So there are other ways of inducing seizures with, for example, drugs that block inhibition, picrotoxin, and that's very reproducible. In other words, you add the drug and you get a seizure, right? Do you think that would be another way to use statistics?
I would say for focal epilepsy like that, I'm very interested to look for ways of focal influence. Because if you give systemic drugs, they affect your area. So let's say you do not have epilepsy, but you have secondary effects. Let's say you cannot drive a car, or I mean, kind of, attention, whatever. Yeah, so for focal epilepsy, I would prefer to look for something that can be applied, not DREADS, at least in the current form, but there's a way. So I could not get the money to do it, but the way is to take caged DREADS, then to use focal ultrasound stimulation to inject them in the blood, to release them where you want, and then to activate them with food. That you can add. What is the name? I don't, but there's a substance you can eat and it will activate DREAD. So it's all becomes possible, but the technology, how do you do it in primates and then in the other species of primates, right? Human being, right? So it might not be all that easy. Yeah.
Okay, well, let's thank Igor again.
Thank you.
And so we're going to break for lunch and come back here in